Welcome to our overview of mRNA vaccine science, where we unpack how a blueprint becomes immunity. In this presentation, we'll trace three core development stages: mRNA synthesis, lipid nanoparticle formulation, and clinical validation and scale-up, while highlighting the underlying biology and strategic choices that make rapid, safe antigen production possible in modern vaccinology. This slide explains how laboratory engineering improves mRNA performance, focusing on codon optimization, modified nucleotides, and designed untranslated regions. These changes boost translation efficiency, dampen unwanted innate immune overactivation, and extend molecular half-life, ensuring enough antigen is produced and presented to the immune system before cellular clearance for an effective response. Here we follow the delivery journey from injection to protein synthesis. Lipid nanoparticles encapsulate and protect fragile mRNA, then fuse with muscle cell membranes to release payloads into the cytoplasm. The mRNA remains outside the nucleus, allowing ribosomes rapid access to translate spike protein, which is the essential first step for antigen production and immune recognition. This diagram shows how ribosomes translate the mRNA into spike protein fragments, which are then processed inside the cell and presented on MHC molecules. That presentation primes both cytotoxic T cells and helper T cells, generating a coordinated adaptive immune response, while secreted fragments can stimulate antibody-producing B cells for broad protection. The slide outlines innate sensing through endosomal toll-like receptors and cytosolic RIG-I receptors recognizing foreign RNA, triggering interferons and cytokines. That early signaling recruits dendritic cells, enhances antigen presentation, and creates an inflammatory context that shapes and strengthens the subsequent adaptive immune response, acting as a crucial bridge between innate detection and long-term immunity. After spike protein presentation, the immune system mounts a dual-pronged response. Immediate activation produces neutralizing antibodies from B cells and cytotoxic potential from CD8 T cells, while durable memory pools of B and T cells form. Those memory cells enable rapid, high-affinity protection on future encounters, offering effective defense without exposure to live virus. This page describes computational antigen design, starting with viral genome analysis to find conserved, surface-exposed regions, then epitope selection, using HLA binding affinity models. The process balances strong immunogenicity with escape avoidance and finalizes optimal mRNA target sequences to maximize population-level efficacy while minimizing the risk of immune evasion. mRNA production begins with enzymatic synthesis from a GMP linear DNA template using RNA polymerase, followed by downstream purification steps like denase digestion and chromatographic separation to remove impurities and short transcripts. Rigorous quality control confirms identity, purity, potency, and integrity, yielding capped, polyadenylated mRNA that meets specifications for safe human use. This timeline outlines the clinical and regulatory pathway. Preclinical studies assess toxicity and safety in models. Phase I uses dose escalation for initial safety. Phase II expands safety and defines immunogenic dosing. And Phase III performs large-scale efficacy trials, leading to emergency use or full licensure, following regulatory review based on infection outcomes versus placebo. Post-authorization surveillance combines real-world data collection with continuous signal detection to confirm high protection, identify rare adverse events early, and guide optimal booster timing. These pharmacovigilance systems complement randomized trials by providing long-term effectiveness, evidence, and support transparent policy adjustments that maintain public trust and vaccine uptake. Next-generation platforms focus on delivery and RNA innovations, such as ionizable lipid optimization, polymer nanoparticles, and self-amplifying RNA to improve stability, lower dose needs, and enable alternative administration routes. Strategic goals include better thermostability, mucosal or intradermal options, reduced manufacturing burden, and broader global access, while preserving safety and strong immunogenicity. The final slide looks to future applications beyond infectious disease. where the mRNA platform can encode neoantigens for cancer immunotherapy, promote regulatory responses for allergen tolerance, or supply mRNA for protein replacement therapies. Together, these directions point to a strategic shift toward personalized nucleic acid medicines that deliver precise, individualized treatments across medical fields.